A researcher modifies a protein so that several buried hydrophobic residues become exposed to solvent. Which observation would be expected? Okay, so first off, right off the bat, before we even read the answer choices, let's just keep in mind that we're exposing hydrophobic residues to water. So what do we expect? Well, we should be expecting a decrease in the entropy of water. We know that this is going to force water molecules to form hydration shells. And then as a result, they're going to be more ordered because they have to form these hydration shells around hydrophobic residues, and the molecules that form those hydration shells won't be able to move as much. So let's just demonstrate. If you have a valine, let's just pretend it's this circle. We know that water molecules are going to have to surround the valine, and that they're going to do so in a very ordered manner. And so if you have two valines, for example, instead of just one, we know that you're going to have less water molecules and then surrounding them in an ordered fashion than if you had two valines drawn separately. Because think about it, this overlap right here is space that water molecules don't have to fill in, that they don't have to surround each valine with. And so if you have two separated valines, this space now isn't filled, and now we have to add additional water molecules that have to be ordered, and so as a result, we'll have less entropy. So let's keep that in mind as we move through the answer choices. Answer choice A, increased ordering of surrounding water molecules and decreased solvent entropy. Well, that makes sense. That's literally what we talked about. If we're exposing buried residues to solvent, we see we're doing this. So we're going from this case to this case. And so what do we see? So we're increasing the order of surrounding water molecules. That's correct. Because now there's more space around each valine that needs to be surrounded by water. And as a result, we're decreasing solvent entropy. And that's exactly what's happening. Since we're ordering more water molecules in hydration shells around the valine, that is going to be decreasing solvent entropy. So answer choice A seems correct. B, increased formation of peptide bonds within the protein. Well, exposing hydrophobic residues to solvent isn't really going to be affecting peptide bonds in any way, because whether the protein is in a solution or in a hydrophobic environment, or whether the pH is one thing or another, or the heat is one thing or another, the peptide bonds are never changing. Remember, peptide bonds are primary structure, and they are not impacted by the environment of a protein after they're already formed. D, increased protein solubility due to exposure of nonpolar residues. Well, we know that nonpolar residues are going to decrease solubility. If you have something that is a hydrocarbon, it doesn't want to mix with water. For example, oil. Oil is nonpolar. And so if you expose it to water, it will not mix. We're decreasing the solubility by adding something like oil or something hydrophobic. And so answer choice C is wrong. And then D, increased protein stability due to greater interaction with water. Well, remember, we're decreasing solubility. And so as a result, we're also decreasing protein stability. Now there's going to be less favorable interactions. We're decreasing the hydrophobic effect. And so that means our protein is going to be so much more energetically unfavorable. It's not folded the way it wants to be. It's not in the environment it wants to be in. And so even though we are having a greater interaction with water, this is making more hydration shells, less entropy. And that is going to be unfavorable. And that's why answer choice D is wrong. A protein complex consists of two subunits. Subunit A, which is 40 kilodaltons, and subunit B, which is 20 kilodaltons. The subunits are held together by disulfide bonds. What pattern would appear under different electrophoresis conditions? Okay, before even looking at the answer choices, let's just think about the three methods presented in all of the answer choices. So we have a native page, an SDS page, and a reducing SDS page. So let's just think about what these three methods are and what we'd expect the pattern to be, or the final pattern to be. Well, remember, a native page doesn't manipulate structure. It keeps structure consistent in a peptide. And so the overall pattern we're expecting is the original structure. And since we know that we have a 40 kilodalton subunit plus a 20 kilodalton subunit that are bound covalently by disulfide bonds, we should end up in a native page with a 60 kilodalton protein. Okay, fair enough. Now let's move to SDS. Remember, SDS manipulates secondary structure. We're going to be manipulating alpha helices, beta sheets, and remember this is by adding a uniformly negative charge to the entire protein. What an SDS doesn't do, however, is manipulate disulfide bonds. Remember, these bonds are covalent. Covalent bonds cannot just be manipulated by adding a uniformly negative charge to a protein. And so again, because the subunits are simply put together by covalent bonds, by disulfide bonds, we still expect a 60 kilodalton product. And that's because we're not destroying the disulfide bonds that are holding this protein together. Yes, the secondary structure might be completely ruined in both subunits, but they're still going to be stuck together and still going to be traveling together on an electrophoresis gel. And now a reducing SDS-PAGE. Remember, reduction destroys disulfide bonds and oxidation forms disulfide bonds. So if we destroy disulfide bonds, that means we're expecting to split up subunits A and subunits B into two individual things. We're cutting those disulfide bonds holding them together, as they're just going to split up. And because they're different sizes, they're going to migrate differently on an electrophoresis. And we know even more that since subunit A is bigger, it's going to travel slower and the band for it should appear higher on the gel. And since subunit B is smaller, it's going to travel faster and should appear lower on a gel. And so now let's look for an answer choice that has exactly the pattern we're expecting. So let's begin with just a native page. We are expecting a 60 kilodalton product. So we know answer choice B is wrong because it says we're expecting a 40 kilodalton and 20 kilodalton product. And we know answer choice D is wrong because it also doesn't represent the 60 kilodalton product we're expecting. So now let's move on. We know for an SDS-PAGE we're expecting 60 kilodaltons. So that knocks off answer choice A because that's not what it has. And then let's just double check that answer choice C has everything we're looking for. So we already went over this, and now for a reducing SDS-PAGE, we get two products, both equivalent in size to the subunits. So that's why answer choice C is correct. A researcher wants to isolate a protein with a pI of 8.5 using ion exchange chromatography. At what pH would the protein most likely bind to a negatively charged column? So remember, ion exchange chromatography is just when you elute some sort of sample through a column that has beads or resin in it that have a certain charge. And so we're trying to find the proper pH to elute our sample in which it will bind to these negatively charged beads. So let's think about that. We know that if we want something to bind to a negatively charged bead, it has to have a positive charge. We also know that the pI of whatever we're looking at is 8.5. And so remember, this is just the pH that makes the protein or makes the sample have a net charge of 0 or be a Zwitter ion. Okay, so let's just think about the relation of pH versus pI. Remember, if pH is less than pI, our sample will be positive. If pH equals pI, our sample will be neutral. And if pH is greater than pI, our sample will be negative. And so we're looking for this case. We want our sample to be positive so that it can bind to a negatively charged bead in an ion exchange chromatography experiment. And so let's look for an answer choice in which the pH is less than the pI. So A, a pH of 10. Well, that's wrong. Don't even have to read the rest of the answer. pH of 7. Okay. That's correct. pH of 8.5, well, that's not less than pI, that's equal to pI. And then D, pH 8.5, same thing, that's equal to the pI, so it's going to be neutral. And so look, even by knowing just the relationship of pH versus pI, you can ignore all this material in the answer choices. You just have to look at the pH values to see which answer makes sense. And so that's why it's super important to have a solid foundation in these relationships.